Hello, dear students. I welcome you all on behalf of Sri Jayendra Rangjone classes. Dear students, we are in a continuation of the chapter that is the cell, the unit of life. Okay. So, dear student, in some of the previous classes, we already covered a difference between the prokaryotic and eukaryotic cell. Then we have also discussed about the cell membrane, cell wall, and we can say endomembrane system in case of eukaryote in detail. If we talk about today's lecture, so in today's lecture, we are going to discuss mainly about the mitochondria and the plastids. Okay. So if we talk about the mitochondria, the term mitochondria is a plural. Actually, singular term, if we talk, that singular term is mitochondrion, and it is not visible unless it is stained under the microscope. So they are smaller in size. Because of this, they are not easily visible with the naked eyes or the microscopic view. So they need to be stained. The number of mitochondria per cell varies depending on the physiological activity of the cell. The number of mitochondria can vary. Typically, if we talk, it is a sausage shaped or cylindrical shaped and having a diameter of 0.2 to 1 micrometer. Average is 0.5 micrometer and length is 1 to 4.1 micrometer. So this is general introduction to the mitochondria. It is saying that this is smaller in size. And if some cells, see, mitochondria is actually considered as a powerhouse of cell. Why it is considered as powerhouse cell? Powerhouse of cell? Because it is synthesizing ATP. So if particular physiological activity of one cell is high, then there is a need of a large number of ATP or more ATP. So the number of mitochondria will be more per cell. But if the activity, physiological activity is less, then there is less need of ATP. So at that time, then you will see that the number of mitochondria are less in that particular cell. And each mitochondria, if you see the structure at a structural level, you will see that it is double membrane bound with the outer compartment and the inner compartment that are divided by the lumen. So we will discuss what are the outer and inner compartment. So first, let's discuss some more point. So we have discussed that number depend on the physiologically activity of the cell. It is known that all the mitochondria, all the mitochondria present in a cell, so all the mitochondria which are present in a cell are collectively called chondriosome. Chondrium actually. Okay. So the term used for the all the mitochondria present within the cell. Usually it is considered that usually plant cell have fewer mitochondria as compared to animal cells. And we will study in the continuation of this lecture that in plants are chloroplast is also there. And that chloroplast has also a capacity of ATP synthesis. So in plant, ATP synthesis is carried out by two organelles, that is mitochondria and chloroplast. But in animal cell, only one particular mitochondria is carrying out ATP synthesis. So they are large in number in animal cell. It is known that in in higher animals, Maximum mitochondria are found in flight muscle of birds. So in flight muscle of birds, you will see that the maximum mitochondria are found. And diameter already we have studied. See, this mitochondria is also named as a powerhouse of cell or ATP mill of cell because it is mainly involved in the ATP synthesis. It is also obvious a most busy and active organelle in the cell, and we will discuss that it is a semi-autonomous organelle. Okay, it is a semi-autonomous organelle. Why it is semi-autonomous organelle? Because it has its own DNA. We will discuss that. Its own ribosome, its own structure, which is independently one look like a one separate cell. So sometimes it is also called that mitochondria is one type of cell within the cell. Obviously, it is not that, but sometimes it is used. So, if we talk about mitochondria, the structure somewhat look like this. Okay, this is the mitochondria. And if we see in the mitochondria, you will see that there is a inner membrane. This blue color inner membrane you can see. Then there is a outer membrane. See, outer membrane is this one. This is the inner membrane. And inner membrane is showing a folding. And this folding are known as what? Cristae. Then you will see within the inner membrane you will find a lumen, and in that or matrix we can say mitochondrial matrix. In mitochondrial matrix you will see the presence of ribosome, granules. Okay, these all things are seen. So it is not clear that what is there in the structural part. So let's draw one diagram. And if we draw a one diagram of the mitochondria, actually it will look like this. This is the outer membrane, then this is the inner membrane. This is inner membrane is showing a folding like this. Okay, like this mitochondria is. If we talk about this is the outer mitochondrial membrane. Then, if we talk, this is the inner mitochondrial membrane. These are the foldings of inner mitochondrial membrane, and this folding are known as cristae. And in cristae, the term cristae is used. The term cristae is used. So cristae is a particularly what we can say here are the foldings of inner mitochondrial membrane. So we can see that mitochondria is covered by a double unit membrane or similar type of membrane which are present and that is covering here. This outer mitochondrial membrane, it is considered as a smooth membrane, whereas this inner mitochondrial membrane folds into several cristae, okay, Now, okay, so these cristae are there, which are the inner folding of this one. Okay. Now, after this is the mitochondrial matrix. If we talk about this region, this is considered as a mitochondrial matrix, which consists of fluid. In this whole part, this is the mitochondrial matrix. And in mitochondrial matrix, if we see, they have their own double-stranded circular DNA. Double-stranded circular DNA. And apart from this, you will see the one more thing, that is a ribosome. And they have their own ribosome. And this ribosome are of 70S type. Okay. 70S type of ribosome are seen. Okay. And their circular double-stranded DNA, if we talk, they are GC rich. GC rich means guanine and cytosine rich DNA is there in this particular DNA. The space which is there between the outer and inner mitochondrial membrane, this space is known as perimitochondrial space. We can say PMS. And over cristae, you will see that there is a presence of several F1, F0, F1 particle. You will see that the several F0, F1 particle are there on the cristae as well as on the inner membrane. So this is a F0, F1 particle which is known as okay. So this is, if we talk, this is F0, F1 particle or oxysome. We will talk about this also detail in the continuation of this lecture. Okay, so this is the thing which is seen. You will see the presence of ribosome and circular DNA, their own circular DNA in the, we can say, a mitochondrial matrix. So the inner compartment is called a matrix. The outer compartment, outer membrane forms the continuous limiting boundary of the organelle. Outer membrane is the one continuous boundary, whereas the inner membrane is showing folds, infoldings, which are known as cristae, and singular is crista. Actually, just I have written plural crista. Okay, that is a mistake. Just okay. We remember this is a cristae is a plural. It should be crista. Okay. Okay, and cristae is a plural. Okay. So the cristae increases the surface area of the inner membrane. And in mitochondria, you will see that the aerobic respiration is occurring. So it is the site of aerobic respiration. We will see that in mitochondrial matrix, if we will study the respiration chapter in the physiology part. And in that also we will study that one Krebs cycle is the thing which occur in the inner on the mitochondrial matrix. Whereas electron transport chain also part of a respiration that also takes place in a mitochondrial membrane. So we will discuss that. So which type of respiration occurs in mitochondria? That question can be asked. We remember aerobic respiration. In aerobic respiration, this mitochondria are involved. Whereas if we talk about mitochondria, they produce a cellular energy in the form of ATP. Actually, just we have seen the F1 F0 particle in the diagram. That F1 F0 particle is involved in the ATP synthesis. And due to the mechanism of ATP synthesis, it is also known as mitochondria, is also known as powerhouse of cell. Then the inner compartment, that is the mitochondrial matrix, and the outer compartment is the what? It is the perimitochondrial space. The inner compartment, that is mitochondrial matrix, also possess single circular DNA molecule. There is a double-stranded circular DNA. Then there's 70S ribosome, and there several few RNA molecules are also found there. Then the mitochondria divide by fission. If mitochondria want to increase in number, they will divide by a fission, and the number of mitochondria will increase within the cell. What are called as powerhouse of cells? The direct question can be asked. You know that mitochondria are there. Okay, so this is the thing. Now, in a structural and all the component, we have seen so many things. So we have to note down some important points. We have initially studied that mitochondria is specifically stained and then they are visible under microscope. Otherwise, they are not directly visible under the microscope. So the question may be asked, by which stain they are stained. So we remember mitochondria are stained by genus green bee dye. Genus green bee. This is a dye which is used to particularly stain the mitochondria. We have already understood that mitochondria is a double membrane bound structure and both the membrane are unit membranes. We have also studied that outer membrane is smooth, inner membrane has folds which are known as cristae. Now it is known that the outer mitochondrial membrane has more phospholipid and the phospholipid which is used here, that is phosphatidylcholine. So this phosphatidylcholine is a phospholipid and cholesterol. So outer membrane has more phospholipid and cholesterol. The phospholipid which is present in outer mitochondrial membrane, that is specifically phosphatidylcholine, that are more compared to inner mitochondrial membrane. Now the phospholipid in inner membrane, that is a type of lipid in inner membrane, is mainly diphosphatidylglycerol and inner membrane have more protein. The protein are more compared to the outer membrane, but outer membrane has more lipid. The space which is present between the outer and inner membrane, we call is it as a perimitochondrial space, and that is the outer compartment, whereas the inner compartment is the mitochondrial matrix. So we can say that outer compartment is perimitochondrial space and inner compartment, if we talk, that is mitochondrial matrix. So such two type of compartments are formed due to the outer inner membrane. Now, if we study further, then we come to know that the inner compartment, or we can say mitochondrial matrix, it is filled with a dense homogeneous substance. It is filled with dense homogeneous substance called matrix. Okay. These two membranes, outer and inner membrane, have their own specific enzymes associated with the mitochondrial functions. When the metabolically active mitochondria have more cristae, we remember, because cristae have a F0 F1 particle which is carry out ATP synthesis. So if it is a more metabolically active mitochondria, then there is a more need of ATP. So in metabolically active mitochondria, number of cristae are higher. Then we will see that there are many electron carriers arranged in a definitive sequence on the inner membrane of mitochondria, which is called an electron transport chain. So we will discuss what is electron transport chain in the respiration chapter, but you should remember the location. Then electron transport system or chain. If we call system, then it is ETS. So this electron transport chain include many electron carriers like cytochromes in a definite sequence. So this electron mitocycle take place in inner mitochondrial membrane. Then on inner membrane you will find a cristae, and over cristae you will see the presence of oxygen on the inner membrane, as well as on the cristae you will see the presence of oxygen. So inner membrane and its folding, that are cristae, have pin-like or pinhead particles called oxysomes or elementary particle. And this oxysomes or elementary particle, if we talk, they are also known as F0 F1 particle or ATP synthase or Fernandez and Moran particle. Okay, so F0 F1 particle, ATP synthase, elementary particle, oxygen or Fernandez Moran particle. It is known as a F0 F1 particle. Now this somewhat look like this. Its structure is somewhat look like this. The structure of oxygen is somewhat look like this. And in this, we will see that this is the F naught particle which is located in the inner membrane. So F naught is the inner membrane, whereas this is the F1 particle, and this F1 particle is the head part which is protruded or extended in matrix of mitochondria. Okay, so this is extended in the matrix, whereas this F naught is located in the particularly inner membrane. Now this head, if we talk, that is oxygen. This oxygen or the head of this oxygen is involved in phosphorylation of ADP, and phosphorylation of ADP will form a ATP. Okay, so it is known that head of oxygen or F1 concerned with is concerned with oxidative phosphorylation. That is formation of ATP by energy of oxidation. So this oxidation actually takes place in the ETS, electron transport system. And from where the energy which is released, that is used in the particular synthesis of ATP, so it is known as oxidative phosphorylation. One more cycle of respiration that is known as Krebs cycle, that is also involved in breakdown of glucose during the aerobic respiration. This is located, its enzyme are found in mitochondrial matrix. So its enzymes found in mitochondrial matrix. Okay, so it is Krebs cycle will also take place in the mitochondrial matrix. Besides this, if we talk, mitochondrial matrix have ribosome, that is 70S type, RNA, and enzymes. So it have its own ribosome, its own RNA, and its own enzymes. So mitochondria is called as semi-autonomous organelles. Okay, so this is the thing which is found in case of this part. Now the DNA which is present is a single molecule and it is a double stranded, it is a circular, it is a naked DNA, means without a nuclear envelope, present in the mitochondrial matrix. And this circular, single circular naked DNA have high GC content. Okay, so mitochondria, if we talk, they have their own proteins or ribosomes actually. So it is known that mitochondrial DNA code for synthesis of code for synthesis of some proteins and rest of proteins and enzymes of mitochondria are synthesized by are synthesized under control of nuclear genes. Means some proteins which are or some enzymes which are required in the mitochondria are synthesized by the mitochondrial DNA. But the DNA which is present in nucleus or a particular nuclear genes that have a capacity to synthesize some proteins in the cytoplasm and send it to the mitochondria. Now we remember this mitochondrial DNA is 1% of total DNA in cell of total DNA in cell, and it is guanine and cytosine nitrogenous base is rich. Okay, and we know that function of this is particularly ATP production. Now if we talk how this particular mitochondria are produced, and we call it as a biogenesis of mitochondria, then it is known that the new mitochondria arise by binary fusion of the existing mitochondria, and the binary fusion will produce a new mitochondria. Then it is known that mitochondria are endosymbiotic in origin from prokaryotic cell. So it is known that mitochondria is endosymbiotic. Endosymbiotic means, suppose initially we know that prokaryote evolved before the eukaryotes, and this prokaryotic cell entered within the eukaryotic cell and it permanently symbiotically or beneficially remained within the eukaryotic cell. So we call it as a mitochondria as an endosymbiotic, and we are comparing it the endosymbiotic nature of prokaryotic cell. Why? Because it has some similarity with the prokaryotic cell, because the type of DNA is GC rich and circular and double stranded. That is the nature of prokaryotic DNA, and this DNA is also in mitochondria is of this type. Apart from this, the 70S ribosome which are found in mitochondria, the similar type of ribosome is found in case of the prokaryote. And the division by binary fission. So this all states that obviously mitochondria evolved from the prokaryotes, and it is a semi-autonomous as well as endosymbiotic organelle. Okay, I hope dear students you understood this mitochondria. So let us discuss few MCQs. The type of ribosome in mitochondria is obviously just we have studied, they are 70S type. The powerhouse in plant cell are, obviously we know that mitochondria carry out a ATP synthesis, so it is a powerhouse of cell. Krebs cycle takes place in Krebs cycle is the one cycle which takes place in the aerobic respiration, and obviously it is found in mitochondrial matrix. The cell organelle which has a self-duplicating nature is self-duplicating nature means which can replicate its own, and it is known that by binary fission they form their copies. So mitochondria is the self-duplicating organelle. Respiratory enzymes are abundant in respiratory enzymes. Obviously, they are because respiration is the main feature or function of mitochondria. So obviously, they are more in the mitochondria. Okay, so here this is all about we can say mitochondria in detail. Now we are going to discuss about the plastids. Okay, so if we talk about plastids, let's discuss them. So plastids are found in plant cell and euglenoids. They are known to seen in the plant cell and the euglenoids. Euglenoid is one type of protist. We know that. And in some algae also this chloroplast of these plastids are found. Now these plastids bear specific pigment, and due to the presence of their specific pigment, they have a specific color and they give a specific color to the part of the plant. And if we talk plastids, plastids are of different type, that is a chloroplast, chromoplast, and the leucoplast. First, we are going to discuss something about the some points regarding to the these pigments. Then we will discuss about this in detail. Dear students, if we talk these plastids, they are found in all plant cell and euglenoids. They are large in size. Mitochondria, what we have studied, they are smaller in size. They are large in size. So they are easily observed under microscope. And so specific staining is not required. And based on the presence or absence of a type of pigment, they are of three types. Just we have discussed that plastids are of three types, that is a chloroplast, chromoplast, and leucoplast. Now, first we are going to discuss about the chloroplast, which is one of the important organelle for our study, because in plant physiology we are going to discuss one chapter that is known as photosynthesis. For to understand a photosynthesis, first we should have a knowledge of this chloroplast. So chloroplast, if we talk, the chloroplast contain chlorophyll and carotenoid pigment. This carotenoid and chlorophyll are the type of pigment. What is the difference between that? That all thing we will discuss in photosynthesis chapter. Chlorophyll and carotenoid are the responsible for trapping light energy. What is the role of them? They trap light energy, they absorb light energy, and carry out a photosynthesis. So their main important function is a photosynthesis. If we see the structure of chloroplast, the chloroplast actually look like this. You can see this chloroplast has a outer membrane, okay, and the inner membrane, okay. So means this is a double membrane bound organelle like mitochondria. Okay, then you will see that the thylakoids are there. Thylakoids are present in the form of pileup, and this pileup of thylakoid is there, which is known as granum. That is a stack of thylakoid. Now, this all thylakoids are formed due to the folding of this inner membrane. Now, this granum, the plural term for granum is grana. This granum are connected through the stroma lamella, and the fluid which is present in this that region represent what? A stroma. And between outer and inner membrane, you will see the presence of intermembrane space as well. So this is the general overall structure. But obviously, we have to discuss them in a proper manner. So if we talk about this chloroplasts, which are important for the photosynthesis, they have variable length, that is 5 to 10 micrometer in length, and width 2 to 4 micrometer, which are comparatively larger than the mitochondria. The number varies from 1 per cell of the chlamydomonas. If we talk about the number is 1 per cell in chlamydomonas, that is a green algae, but it can be up to 20 to 40 per cell in the mesophyll cell of the higher plants. And if you talk about this, majority of chloroplast of the green plants are found in mesophyll cells. So this is again the structural part, and in structure you can see the outer membrane, inner membrane, the thylakoid. The thylakoid pileup is a granum, and this is the stroma, and the stroma lamella is here, which is connecting them. So like mitochondria, the chloroplasts are also double membrane bound, just we have discussed. The space limited by the inner membrane of the chloroplast is called a stroma. So the space which is present within the inner membrane, it is known as stroma. And the number of organized flattened sac called the thylakoid. Thylakoid are the sac-like structure which are present in the stroma. And the space which is mainly present surrounding to this thylakoid, that is known as what? A stroma. But when thylakoid are found, they are not found alone, they are found in a group sometimes. So this arranged stack of this thylakoid called a pile, it look like a pile of a coin, it is known as grana. A granum is a plural term. So you can see this is the grana. And you can see here, this is the outer membrane, this is the inner membrane, this is the stroma, this is the thylakoid. See, the thylakoid which is present in this granum, it is known as grana thylakoid. Okay, or we can call it grana lamella. Whereas this is the stroma thylakoid actually, which connect to granum. And this stroma thylakoid can also be called as stroma lamella. So if somebody asks what actually mean by lamellae, so lamellae actually a term for the thylakoid, and we can see here that two type of thylakoid are seen. One is grana thylakoid, which are in a granum or a stack-like arrangement, whereas the two grana are connected by the stroma thylakoid or a stroma lamella. So what is stroma? It is a space limited by the inner membrane of the chloroplast. And what are thylakoid? They are the number of flattened membranous sac-like structures. And what is the grana? Nothing but it is a thylakoid arrangement in a stack. So flat membranes tubules called the stroma lamella connecting the thylakoid of the different grana. And the membrane of the thylakoid encloses a space that is called a lumen. So lumen is the innermost space which is covered in the inner membrane. You can see here. This is the we can say here they have not this stroma they have written. Okay, but this is the thylakoid, and within the thylakoid this space is found. This space is known as lumen. Okay, this space is known as lumen within the thylakoid. And within the chloroplast you will see what? A stroma. So here you can see this is the actual diagram. Important again, outer membrane, inner membrane, granum, lumen. Lumen is the what? When we cut this thylakoid, the space you will see this in the particular thylakoid, that is a lumen. So within the lumen, within the thylakoid you will see the lumen, and this is the stroma. Okay, so it is more correctly it is shown in another cross-sectional diagram. The stroma is the outer membrane, this is the inner membrane, then the stroma, this is the thylakoid. And this thylakoid form a stack that is known as the granum. And when we cut a granum, you will see the lumen, a space in the thylakoid. That space in the thylakoid is known as lumen. Okay, so this is the space in the thylakoid. This yellow color, it is nothing but a lumen thylakoid. So the stroma contain enzymes, and this contain enzyme which carry out a dark reaction of photosynthesis, and they are required in synthesis of carbohydrate and protein. Okay, so carbohydrate synthesis occur through the dark reaction in photosynthesis. Apart from this, they have ribosome, so they can carry out protein synthesis. Okay, so photosynthetic pigments are present in the thylakoid membrane. Yes, they are present in the thylakoids, but actually they are present in the thylakoid's membrane. And these pigments are found in group. So pigments are responsible for trapping light energy for the photosynthesis, and mainly we know that chlorophyll is there. So the chloroplast also contain ribosomes, 70S type, small double-stranded circular DNA. Okay, they have the ribosome which are 70S type, similar to the mitochondria. But in cytoplasm, you will see 80S ribosome. They have a small double-stranded circular DNA which is similar to the what? Mitochondria. Now, when we see the ribosomes in the stroma of this chloroplast, you will see that the chloroplastic ribosome are found, which are small in size and they are 70s. Prokaryotic type are there. Whereas when we see the cytoplasm of the cell, they have a 80s eukaryotic ribosome. So ribosome of the chloroplast are smaller in case of which because they are 70s and prokaryotic, whereas the cytoplasmic ribosome are eukaryotic and 80s type. Okay, so this is the thing with respect to the what chloroplast. Now we remember some important points with respect to the chloroplast, that each chloroplast has 40 to 60 granum. 40 to 60 granum are found. One more important point you should remember, that the stroma lamella or stroma thylakoid or stroma lamella, it is also called fret channels because they are connecting what to granum. So they are also known as fret channel. And these are the flat membranous tubuli connect the thylakoids of the different granum. Now we remember approximately 250 to 400 molecules of various photosynthetic pigment constitute or form one quantazome. So such several quantazome can be present within the thylakoid membrane. Now if we talk about the thylakoid chloroplast, the chloroplast have some enzymes, and in chloroplast the stroma of chloroplast contain enzymes of Calvin cycle, or we can say the dark reaction. Calvin cycle is the C3 cycle. Why it is known as C3 cycle? That all thing we will discuss in the photosynthesis chapter. And they have their own small double-stranded circular DNA. They have their ATS ribosome. That all things we have already discussed. Now we remember one more important point, that the outer membrane of chloroplast and mitochondria, which we have already discussed, have large sized proteins known as porins, and these porins are that much large size that they can allow the transport of some small proteins as well. Apart from this, if we talk about chloroplast, we know that chloroplast have their own genetic system like double stranded circular DNA, RNA, then protein synthetic machinery, then like ribosome, enzymes. This all thing they have. Okay. So they have this all structures and their own materials. So they are called, so also called semi-autonomous organelle of cell. So it is the second semi-autonomous organelle. First was the mitochondria. Okay, so this is the thing which is carried out by the, so they have their independent machinery, they can carry out their own functions. Okay, so because of this particularly, they are, we can say, are known as semi-autonomous organelle of cell. After this, dear students, if we talk about function, the important function of chloroplast is to carry out photosynthesis. And we know that in photosynthesis, two reactions take place: light reaction and dark reaction. Okay, so RXN means reaction. So this light reaction involve a photosynthetic pigment, so it occur on membrane of thylakoid, and this dark reaction take place in stroma of chloroplast. Now after function, if we talk about this biogenesis or its synthesis of chloroplast, then first they getting synthesized or originate from proplastids. So proplastids are the immature plastids, we can say. And second, from binary fission of pre-existing plastids. Okay, so from the pre-existing chloroplast, this chloroplast can divide and they can, we can say, form a new chloroplast. And obviously, the chloroplast are also endosymbiotic in origin, like mitochondria, because it has its own DNA, its own RNA, its own proteins, its own enzymes, its own ribosome. All this they have their own. So because of this, it is also a, we can say, endosymbiotic in origin. Okay. So this is what about the chloroplast. Now after this, we are going to talk about the some another component like chromoplast, which is the second type of plastid. When we talk about chromoplast, in the chromoplast, fat-soluble carotenoid pigments like carotene, xanthine, and others are present. So we will see that here in chromoplast, carotene, xanthophyll-like pigment are present, and which are fat-soluble. This fat-soluble pigment, or we can say the carotenoids, gives a yellow, orange, and red color to the parts of plant. They are responsible for giving the disease different color because they are colored. Now, the chromoplast gives color like this. Okay. So if chromoplast gives such color, it is known that They are giving a red, yellow, and orange color. So in this, it is considered that chlorophyll, which is a green color pigment, they are either absent or occur in very less amount, because of which the carotenoids are dominant in the chromoplast. This chromoplast mainly occur in pericarp of fruit and petals of flower. And in petals they are present, because of this they give a different color, and due to which the different insects are attracted toward the flowers. Now we know that tomato are red color. So it is known that red color of tomato is due to red pigment, that is lycopene, which is the basic unit of carotene. This chromoplast occur in petal and give them color. Okay, so one pigment, anthocyanin, it is water soluble pigment that is found in cell sap of vacuole. Actually we have studied, and that is also one pigment, and that is found in some particular chromoplast sometimes, but not mainly. So the chromoplasts they also have this anthocyanin sometimes. So this is the what with respect to the, we can say, the chromoplast or colored plast. Now after colored plast, next we are going to talk about a leucoplast, which is a colorless plaster. And this leucoplast are the colorless plastid of various shape and size, but the main function of them is to store nutrients. So based on the type of nutrient they getting stored, the leucoplast are of different type, like amyloplast, elaioplast, and aleuroplast. So if we talk about these three, that is amyloplast, elaioplast, and aleuroplast. Amyloplast, if we talk, they look like this. And this amyloplast, it is known that they store carbohydrate. They store carbohydrate, or we can say that stored carbohydrate in the form of starch. And if you take example, an example is potato. Okay, that has a rich, it is rich in amyloplast. After this, there is another type of leucoplast, that is elaioplast, and this elaioplast look like this. And another one is the eleuroplast. If we talk about elaioplast, it is known that this elaioplast, see, the term is oil is coming. Oil. If you repeat, you if you, for remembering, oil is coming. So you can remember that elaioplast, or it is just for the remembering, oil and fat. Whereas if you talk about eleuroplast, this eleuroplast store protein. Okay, because we will study that eleuron layer is found in maize, which is a proteinaceous in nature. So eleuroplast term is proteinaceous in nature. Okay, now it is known that pigments and lamellar or thylakoid structures are absent in leucoplast. And this leucoplast generally occur in non-green and underground plant part. Underground plant part like ginger is there, potato is there. You will see that this particular different type of component. And it is obviously considered that different type of plastids may transfer from one form to the another form. So if see this leucoplast are there, they do not have a pigment, but if they develop a pigment like chlorophyll, then it become a chloroplast. And if they have a colored pigment, then it can be a chromoplast. In chromoplast also, if they develop a chlorophyll in a dominant form, then they can convert it to the chloroplast. So this chloroplast or on this are can be transformed from one to another because their genetic material is similar. Okay, dear students, so that is about the what leucoplast. So amyloplast, we have stored that they store starch, oil and fat by lyoplast, and lyoplast store protein. And that is the example is seeds of pulses, and in castor lyoplast is same. So if we talk about some MCQ, the number of chloroplasts in chlamydomonas, we have studied that number of chloroplasts in chlamydomonas cell is 1, and chlamydomonas is a single cell. Yellow and orange colored petals and fruits are due to obviously that are due to the colored pigments which are found in the chromoplasts. Rubisco is present in. Rubisco is the full form of Rubisco is ribulose 1,5 bisphosphate carboxylase oxygenase, means it has the capacity to bind with the carboxylase as well as oxygenase, and the CO2 fixation or dark reaction take place within the stroma. So Rubisco is present in the stroma region of the chloroplast. Which of the following plastids are not exposed to light? It is known that actually leucoplasts are not exposed to light, because of this they are colorless. The plastid that give orange and yellow color to flowers and fruit are the obviously the orange and yellow color are given by the chromoplast. Okay, now after this we are going to discuss a ribosome and the cytoskeleton part. Okay, so before discussing ribosome and we are now thorough with the mitochondria and the plastids. Okay, thank you. In next class we will continue this ribosome and the cytoskeletal structures.